Angela Black, welcome to the Anatomy and Clay Learning System podcast.
I'm happy to be here.
It's great to have you on. I mean, I first connected just based on a Twitter photo I think you posted, and reached out to you, I believe, a couple of weeks ago. And we appreciate your featuring your students and some of their work with Anatomy and Clay.
Yeah, actually, I found a Facebook group of teachers who use Anatomy and Clay in their teaching, and I joined that.
That's what it was. That's
right.
what it was. I knew it was social media somewhere, but yeah.
Yeah, that was actually, I was really excited to find that group and to have the opportunity to kind of share ideas and things that work and don't work with different people.
Yeah, great. Well, I want to talk to you a little bit about, of course, what you're teaching. And you're at the University of Maryland, correct?
In the Department of Animal and Avian Sciences.
Right. And we'll get into what you're teaching now and how you incorporate anatomy and clay. But can we just start out a little bit of personal background? When you were young in high school, or when did the whole science idea or teaching idea first take hold? Were you a science kid?
Yeah, I was a science kid. I grew up around large animals, agricultural animals in the Intermountain West area. And I wanted to be a vet, like a lot of kids starting around 8 years old or so. So yeah, I did all the science stuff in high school. In college, I went to the University of Utah, and my first introduction to comparative anatomy was with an amazing professor named Dennis Bramble, who has written some of the key books on functional vertebrate morphology, Hildebrand and Bramble. And I took every class I could take in the biology department with Dr. Bramble. I just loved it. You have to remember, this was in the mid 80s, so you couldn't just search something easily on a search engine and get all this information. To access that kind of information, you had to go to the library and search and copy, or you had to find someone who knew it. So to find someone who could teach me about comparative morphology, vertebrate morphology or functional morphology, was just so exciting. I remember a class project that we did, and I rode horses at the time. And he asked me, he was actually kind of pioneering some work on the idea that in quadrupeds, locomotion and ventilation are coupled so that they help one another. So that when an animal kind of rocks back when they're galloping, say a horse, the diaphragm with that great big liver kind of attached to it rolls back and pulls, and it creates a negative pressure within the chest, draws air in. When they go forward, land on their front legs, again, it acts like a big piston that pushes forward, pushes air out. I was mesmerized. I loved it. And I just enjoyed it. I loved the idea that, our quizzes and things would be sitting out under the trees on the campus, and he would give me a bone and say, okay, tell me everything you can tell me about this animal from that bone. What did it do for a living? What did it eat? How did it defend itself? Was it a runaway fast animal, or was it a stand and fight animal? And I loved learning how to look at a humerus or a femur or, a skull, and learn about that animal. So that's really where my love of anatomy started, was as a biology major. And then I went on to vet school at Colorado State. We studied anatomy and physiology our entire first year, along with some other topics. And I fell in love with the anatomy lab, this huge open space with just unbelievable specimens at my fingertips, and I loved it. I had great anatomy professors there, and it just really continued. I wanted to be a large animal vet, so I took that track, and I went on after vet school to be a large animal medicine and surgery intern at Washington State University's vet school. So, pardon me, I saw clients, saw patients, large animal patients, and then I also worked with fourth year students and taught fourth year students. And what I found during that year was that my favorite days were the days when I taught, which was completely unexpected for me. I had always thought, I'm going to be a large animal vet. I'm going to be an ambulatory vet. That's what I want to be. It would never have occurred to me to want to teach, ever, ever. I'm actually very introverted by nature, and the idea of teaching absolutely terrified me. So, but I remember a day kind of realizing I'm happiest on the days when I'm planning a lesson and figuring out how to, present information and how to teach. And that led to me going on to study for a PhD in animal sciences at Rutgers. And I studied nutritional physiology and bone growth. So I was looking at the effects of nutrition and exercise on the development of bones in young animals. And from there, I went on to be a research fellow, a postdoc basically at the National Institute on Aging. And there I studied bone again. I was looking at aging bone instead of growing bone, but aging bone in non-human primates as they went through menopause and into the postmenopausal period.
Wow.
Yes, I like school, clearly. I love an academic research kind of an environment. I started teaching here at the University of Maryland in 2005. This is my 17th year teaching domestic anatomy of domestic species. I teach the lecture and the lab. I've tried lots of different ideas, different teaching techniques and models over the years. And I just happened to come across Anatomy in Clay. I think it was in 2013. I want to get to that. I want to get to that. And that's, but that was fascinating, absolutely fascinating. Can I just ask you, I'm just personally interested, and I think others might be, your focus on aging bones and the science there. I'm just wondering if you can give us a couple of minutes on what should we know about bone health and taking care of our bones, and what good information could you pass along and sort of walking around knowledge that us folks should know? This is interesting. Well, let's see. For growing bones, you really have to think about evolution, what we were meant to do, right? We're meant to move. We're meant to move a lot, and different kinds of movement. Primates were originally brachiators, up in the trees, and then humans really became superb runners. As true bipeds. Actually, a lot of Dr. Bramble's work is on the evolution of humans as superb and long distance runners. So I think moving for young animals is just, and young, kids, animals is really important. A lot of times we raise dogs and horses to not be as active as they really should be, and people, and allow them to put on too much weight, which isn't good for their joints. It's not the way the bones were meant to grow. I think a lot of the problems that we do see in young dogs and horses and children too is from not just the bones being weak themselves, but the muscles that support them and the ligaments. If the muscles are not strong, it can't support the joints, then we tend to see more, joint problems, kind of the weekend warrior thing when we go out with our dog and play Frisbee, or take our horse on a long trail ride and they're just really not up for it. Just like if we go do something, we really suffer for it on Monday. In
Right.
terms of aging, I was actually on a really interesting study where we were looking at what was then called calorie restriction. Now that's really kind of more interpreted as intermittent fasting.
Right.
So the idea was that, and it's been known since the 1930s, that from everything from yeast to fruit flies to other insects, worms, all the way up into the vertebrates, and the study I was on was in non-human primates, that when you restrict the caloric intake by 20 to 30% consistently, not the nutrition, but the caloric intake, that those animals live about 30% longer, and they
Oh,
have
wow.
fewer chronic diseases. So the study that I was on was looking at a group of rhesus macaques starting in the 80s all the way through their lifespans, watching them age, studying the development of diabetes, cancer, heart disease, osteoporosis, osteoarthritis, and watching the females go through menopause and looking at what happened with their bones if they were calorie restricted. And because that was the concern. The only concern with calorie restriction was, we know that in humans, if you have a small, frail skeleton, you are more likely to get osteoporosis.
Hmm.
So there was that concern. What we actually found is that the animals, since they had very good, their diets were super supplemented, right? So they were not malnourished in any way, shape, or form. They just took in fewer calories. That their bones were smaller, but their bodies were smaller. And so their bodies weren't putting the same biomechanical forces on the bones as if they had these bigger, heavier bodies. So we did not actually, and I left before the study was completely done. My postdoc was over. So I think that data still hasn't been published, but we did not see a deficit or a definite problem with the calorie restriction. And we definitely saw less osteoarthritis in those animals.
Wow. That's really interesting. Really interesting.
It really was. And we definitely know now that the more active you are as you get older, the stronger your bones are and the stronger your muscles are that support your joints. So it's all about staying active. We are an active species. We're meant to move.
So good old common sense rules again.
I'm afraid so. Yeah. And it turned out that the calorie restriction probably wasn't the thing. It was probably that the animals who were eating 30% less actually were eating their food faster, and they were going for longer periods of time when they were fasting. And so it would probably was more of a calorie restriction. Intermittent fasting are very tightly correlated.
Yeah. Good. Thank you for that. So let's go to 2013. I think you said it was when you first encountered Anatomy in Clay, roughly.
Yeah, I was just looking for ideas on how to teach anatomy, different approaches. I like things to change. I know that our students change over time. The students that I was teaching in 2005, 2006, we have a different population of students. They're much more tech savvy. They have different experiences in high school. The needs are different, and I like to keep the class updated. So I was just looking for different ways. And so I thought, well, let me look into how they're teaching anatomy in medical school. And I found Anatomy in Clay. And I was just delighted. And I found the Canican, which is the canine model, also the equine model. But I figured the dog was a better basic model for me to start with.
Yeah. And how did you first start incorporating it into your teaching and learning, and how has that changed over the last 9 years?
Anatomy in Clay was really great. They actually let me borrow several models. I think it was 6 models. And it was just for the one semester. And I surveyed students and said, this is a new method of teaching anatomy. They've been doing it in humans quite a bit, and I explained it. And so I actually asked students if they would be willing to be part of a subgroup that used the ANATOMYINCLAY method, and so that it wasn't forced on anyone. And I learned some interesting things. It was not the best approach because at the time I was using preserved cat specimens and having students dissect in groups of 3 or 4. And what I had with the ANATOMYINCLAY students is I had them kind of look at a dissected specimen, but not really study it all that much because I didn't have the time. We also changed the course so that my lab time is longer. So now I have a longer lab to allow them to have the time. And so we kind of ran into problems with the time to build. And the students who were willing to do it were very patient, and they really tried, and they did a good job. But what I learned from it overall was they really needed some background information first. They really needed to know the anatomy from the dissected specimens first before they built. That's how I felt about it. Now, I know other people who use ANATOMYINCLAY feel that building first and then looking at the specimen is a better way. In my experience, it works better to have the students study dissected specimens first really well and then move to the clay. So that was an interesting year. I kept one of the models, I paid for one of them. And what I did is the next several years, I would build the model week by week as they dissected the cat models, the cat specimens. So they could dissect and study the cat specimens, and then they could come and look at my model that I was building in front of them. And then actually COVID made me kind of rethink the whole class again, because I thought very carefully about how I saw students, what were they doing when they were dissecting? And they were in groups of 3 or 4. And really what I saw were a lot of students, usually 1 or 2 students were actively involved in the dissection, and usually a couple more were on their phones. And I just thought, I'm not engaging everyone. So I thought about that, and then I had to teach anatomy during COVID online. So we turned the whole lab basically into a production studio, and we learned how to video and photograph, and we took great photos, and we taught it online. And the students really did learn quite a bit, but that made me start thinking, well, maybe having them all dissect a lot is really just teaching them how to dissect. Maybe it's not actually teaching them the anatomy. And the more I thought about that, the more I thought, yeah, actually, that's what they're doing. They're spending a lot of time picking off fascia and asking where to cut, but are they really studying the anatomy? And do they need to know how to dissect if what the class is supposed to teach is anatomy?
Right.
So, go ahead.
Yeah.
So I kind of rethought it, and last fall, I was able to buy 6 more models of the Canakin models. And we restructured the lab so that my graduate TAs and my undergraduate TAs, I have a graduate TA and 2 undergraduate TAs in every lab section that has between 18 and 24 students in it. So I have a lot of support for the students in the lab, which I think is important. And we divided it up so that we actually dissected the specimens ourselves. So we dissected like 8 cats, and we would do the weekly dissections really well, nice and cleanly, so everything was really clear. The students would come in, they would get a dissected cat specimen, depending on what we were doing that week. They would have lists of the muscles that they needed or the structures that they needed to identify. And then we have iPads with Apple Pencils at every table, and they had to photograph the specimens and then label them. They could either use labels on the specimens themselves and photograph, or they had to photograph and then label the photographs with the Apple Pencil. And that would be a group participation assignment that they could then upload onto our learning management system. And they would then have those photos, you know, that they had access to on ELMS to study from if they wanted. And it was turned in as an assignment. So then when they're done with that, then they take their Canakin. And we divide it so that 2 people are working on one side of the Canakin and 2 people are working on the other. And they each have a fairly short list of, I think it's usually 7 muscles, if they're doing a muscle lab, that they each have to build. So in groups of 2 on one side, one person is reading the lab manual and saying, okay, you're going to be building the pectoantebrachialis, and this is a thin strap-like muscle, and it is going to originate here and it's going to insert here. And the other person will be actually doing the rolling out and the cutting and the placing of that muscle on the Canakin. And then from week to week, they switch back and forth who does the reading and describing and who does the building. And then when both sides are done with their lists of 7 muscles, because usually each muscle lab includes 14 muscles, then they have to get together and they have to teach each other the muscles they just built. And then they have to photograph them again with the iPads, label them, and turn those pictures in online as their group participation points.
Boy, that's a thorough process.
It is. And I mean, you have to keep them moving. And I think that's one thing that sometimes they don't like, is that there's not a lot of downtime. I mean, they come in, they take a gross anatomy quiz, and then they get their specimens, and they identify, they photograph, they label, and then they get their Canakins, and then they build. We have a 3-hour lab. But wow. Each 2 people build 7 muscles, which actually I think is perfectly reasonable if they're paying attention and have watched the, I actually post photographs of myself building that lab so they can look at that before they come to lab. And they have a manual that I created that lists the muscles, their origins, their insertions, their actions, the order in which to build them, right? From deep to superficial, and has pictures. So they have an idea of what they're supposed to look like.
So as you go back to that original observation that when you're dissecting, you might just be learning to dissect and not so much about the anatomy, what are your observations now? And are they
this was that first year that I did this. Their scores on their gross anatomy quizzes were higher, significantly higher. And I haven't incorporated the models into the gross anatomy quizzes. I've only used the specimens, but I may actually start putting in a few models with some very clear, you know, like the latissimus dorsi, something that's very clear. They should understand, you know, origin, insertion, and they have to know the actions. And we try to talk to them about, you know, making the lines on the muscle between the origin and the insertion, so that they think about, if this muscle contracts, what does it do? I don't insist that they memorize the origin insertion, because, you know, I don't want my car egged. I did that the first year I ever taught, and it was a bit much for a 200 level anatomy class. I think it's a lot of material, and we're doing comparative work too. So I don't insist that they know origin and insertion, but they have to place the muscles on the origin and insertion. And we spent 4 weeks studying the skeleton, studying comparative skeletons. So they know that skeleton pretty well before we ever start muscles or other structures.
Yeah, wow. And you use anatomy and clay in the comparative work as well?
No, because I only have the canakins.
Yeah.
So we don't really do a lot of comparative muscle. I do a lot of comparative skeletal work because I have a pretty big collection of skulls and bones from lots of different species. So we talk about, what can you learn from the skull? And you look at the skull. We have from all the domestic species, a lot of wild species too. I have a lot of articulated skeletons. I actually live on a sheep farm. My family raises sheep, and we have a very hot compost pile. So sometimes if somebody who I know a lot of farmers, and they'll say, "Hey, I have a goat that just died. Do you want the bones?" A hot compost pile is a really great way to clean those bones, and I've learned how to articulate skeletons because they're very expensive to buy.
Yeah.
So I've learned how to clean them. Although now I'm really excited about the possibility of 3D printing bones because they would last longer. I think they would be tougher. When you drop an older bone, it shatters.
Yeah.
So
Yeah.
I kind of love the idea of being able to scan all of the bones in a dog skeleton or a horse skeleton and then articulate it or not. And then if somebody knocks over something over and it breaks, I can reprint a bone. So I'm kind of playing with that idea now.
That's great. Wow. So what would be your comment to somebody who's just looking to get anatomy and clay learning system incorporated in their college level teaching? Do you have any suggestions or advice for that person who might be a little bit
I think the biggest challenge is the cost. It really is. I only have 6 models and I teach between 80 and 100 students a semester.
Yeah.
So that means that 4 students are assigned to 1 canakin per lab section, 2 on one side, 2 on the other. And at the end, they have to pull everything off and clean down that model with the magic erasers, put all the clay away. So we make the most of the 6 that we have. I wish I had another 6. I think it would make it a lot easier. So the cost is one thing. So securing the funding, but so the first time I ever built all of the muscles on that 1 canakin, I was astounded at how much I learned. I finally understood certain muscles that I had never truly understood before. I just hadn't.
Do you remember which ones by any chance?
Yes, and it's not a really complicated set of muscles. It's kind of embarrassing. It's the scalenes of all things. In a human, I think it's pretty simple, but in a quadruped, I knew the kind of turkey foot, the three scalenes, and in a dissection, I had just seen kind of the origin of those 3 strands of the scalenes. And I really didn't have a sense of where they went. I kind of knew what the insertion points were, but when I built those onto the model, I was like, oh, that's what that and the serratus ventralis, that one totally shocked me. I mean, that is a huge muscle in a quadruped. It's a really important thoracic sling kind of muscle, and it's enormous. And the students, I notice, are really astounded at this big, triangular muscle and how wide it is. It goes all the way from the cervical vertebrae back to the ribs, and building really, it solidifies where those muscles are in a way that I just don't, unless you can dissect down to the point where all you can see is that one muscle, I just don't think you can understand it as well. Also, the supinator, the pronator teres, in animals, we call it the oblique carpal extensor or the abductor pollicus longus, those really deep muscles on the forearm of an animal that can pronate and supinate. seeing those on the Cannikin made it so clear in a way that it was never as clear on a dissected model.
That's great. That's great.
So I know this is the best way. I
Yeah.
know it is the best way to understand anatomy. I really do. The comments this past year, some students found it was too much. They would actually say it's too much work. It's too much work. It takes too much time. It didn't really help me that much. Well, but the scores were better. So it did.
Yeah.
and they just don't know, you know, they're mostly sophomores. So they don't really know that if it's really helpful or not.
Yeah.
And I know it really is. So what else would I tell them to do? I would tell them to totally build it themselves first.
Right.
And write down, create the best order of how to build every single muscle. Because dissecting, you're going from superficial to deep. When you build, you're going from deep to superficial. So you have to flip everything. And if you're going to create lists for different groups of students to do, you have to make sure that those lists work together, you know, to create all the flexors and extensors of the forelimb or whatever. So you want these kind of cohesive groups that they're building. So definitely do it yourself first, and keep really clear records and create a very clear step-by-step manual to teach them how to do it. And understand they're not all going to be pretty. They're just not. I mean, some people have some artistic ability and some just don't, and that's fine. It's the process of building. It's the process of creating this shape, this triangular shape or spindle shape or flat shape or whatever, and then saying it goes from here to here, and when it contracts, it does this. So they don't have to all be pretty. And if you demand that everything is pretty, you're going to have a lot of frustration.
Yeah.
Use the time well. Make sure that you don't overload them with doing too much in a short period of time. It creates frustration.
Yep. Yep. Very, very good. Wow, this has been terrific. Angela, thank you so much for.
Oh, and scrape it up off the floor before it hardens. Because if you wait till the end of the semester, you're going to be on the floor with a razor blade trying to get all that clay off the floor. I still haven't figured out exactly the best way to control the clay on the floor. The tabletops and the models we've got, but cleaning it up weekly really does help. You make it fun, and I think you have to really sell it to them. It's often a new idea. Some of them are like, this is so cool, I love this, and they jump right in. Others, I had a few comments about being in preschool again. You know,
Yeah, right.
Yeah, I did.
Play. Yeah.
Yes, it was play. And I actually play in class quite a bit. I will have students play games in class because I think when the mind is relaxed, you learn better.
Yeah,
I really do. And I think the more relevant you can make anatomy to people's lives. So what I tell them is the age of the sage on the stage learning is done. It's over, right? Because anyone can go online and learn anatomy.
Right.
There's no information that I can give you about the details of anatomy that you can't find online.
Right.
But what I can give you, what I can teach you is how to use that information to solve problems.
There you go.
If you're going to be working with living things. And so I flipped my lecture class. I have learning objective videos that the students watch beforehand. Then they take a short quiz before they come to class. And then we spend the class time. Sometimes we spend it drawing. So I will use an iPad and they can use their own devices, or they can use paper. I'll provide paper, and we draw structures, which again is sort of like the anatomy in clay, because what your hand is doing, there's a connection between the hand and the brain. It's just vital. it's one of the reasons I want them to take notes by hand, and I will not provide slides. Because there's something important that happens when you are writing, not typing, but writing and drawing, that cements that information in the mind. And I don't understand what it is, but I've done it long enough, and I know for myself that that's what works.
Well, you sounds like I might have slipped you $1 to say something along the lines of the Anatomy and Clay, motto, but the motto is the mind cannot forget what the hands have learned. So that is
That's right.
exactly.
That's right.
Yeah.
That's right. But I do see it. That's actually why I do the learning objective videos, where there are these 12-minute videos, 12 to 15-minute videos. They have to watch 2 or 3 before they come to class. And they're supposed to take notes on those videos. And those are their class notes. And then we use those class notes in class. I want them to take notes. I don't want them to type, and I don't want them to just print off slides, because there's something important about writing the information down. And then what I really recommend that they do is, after they take notes, that they create an outline and organize those notes. And that they often don't see those activities as studying, but that's studying. It is. Creating study sheets. It's not the study sheet that is important. It's the creation of the study sheet.
Very good. Angela Black, thank you so much for your time today. We can tell your students are very fortunate to have you and your enthusiasm for anatomy. And we appreciate all your insights today, and wish you well down the road.
Thank you. I've enjoyed it, and I love the product, and I hope to be able to gain some more models, and maybe some more comparative, maybe do some of the equine model work as well. So I think it's a wonderful product and a great idea.
Thank you. Wish you well.
You too. Thanks.